My name is Leif Richardson. I work for the Xerces Society for Invertebrate Conservation, and I lead the California Bumble Bee Atlas along with Rich Hatfield, my colleague here. And I'm going to briefly introduce myself and tell you just a few housekeeping things, and then Rich can introduce himself. So I'm an endangered species conservation biologist with Xerces, and I'm based in Southern California, although coming from you today from Santa Rosa because I'm traveling to do bumblebee work with you all in the field. And I have been doing bumblebee work for quite a while and bee work in general also. So my focus is currently on conservation of bumblebees, and the Bumblebee Atlas is a good example of that kind of work that we do. Housekeeping. We have a chat and we have a Q&A. I'm just looking for it. I'm actually not. Oh, there it is. Okay. If you have questions while we're speaking, I'm going to talk, and then Rich will talk, and then I will finish. If you have questions, please type them into the Q&A, and we will answer them, either typed up answers or we will answer verbally at the end of a module. If you want to use the chat, that is for saying hi to everybody and communicating with others who are in the room, but we're not going to take the questions there. So please put them in the Q&A rather than the chat. This is being recorded, just so you know. We probably aren't going to do anything with the recording, but we do sometimes post those to YouTube. And if there are questions we don't have time to answer tonight, I will follow up with answers as appropriate, because sometimes it gets out of hand. We just we don't keep up with all of the good questions. So with that, I'll turn it over to Rich to introduce himself.
Thanks, Leif. Yeah, just also one more housekeeping stuff. There is a closed captioning available. It should be an option at the bottom that should be turned on for you. So if that is helpful for you, please use that resource. It will only show up for you if you click it, so don't feel like you're going to impact the whole world. But feel free to use that resource if you need it. My name is Rich Hatfield. I'm a senior conservation biologist for the Xerces Society. I lead our bumblebee conservation programs. While I got my start working with bumblebees in the state of California, I'm currently based in Portland, Oregon, and I focus on a whole host of bumblebee conservation issues, but most of it is related to these Bumblebee Atlas projects. I started a project in the Pacific Northwest 7 years ago now called the Pacific Northwest Bumblebee Atlas that was kind of the impetus for all of these projects that we now have running in 20 states. So I'm lucky enough now to get to work with Leif on this project and with a team of a number of other people on other projects, which is delightful. And with all of you, which is even more delightful. I feel like, especially in these times that I find challenging, at least, that it's really heartening to be in a room full of people talking about issues that I really care about. And I'm just really thankful you're all here tonight. So thanks for being here, and I'm looking forward to this. For this first session here, I'm going to turn off my camera and be in the background and answer your questions and chat with you. So enjoy. Thanks for being here.
All right. Thanks, Rich. Okay, I'm going to share my screen and we will get started. So yeah, Rich, can you see a title slide?
Yep, it looks good.
Great. Okay. All right. So thanks for being here. This talk is going to be 2 hours with the two of us, and there are 3 modules. The first is an introduction. Then you'll hear from Rich about the methods, and finally you'll hear from me about identification of bumblebees. You heard from Rich about the fact that there are other Bumblebee Atlas projects at Xerces. And in fact, we're working around the nation in a lot of other states. And so you'll hear us refer a bit to some of those other projects, even though this is a very California-specific training tonight. Before I get started, I want to just tell you a little bit about our organization, the Xerces Society. So we protect wildlife through the conservation of invertebrates and their habitats. We organize our work into these programmatic areas around native pollinators, endangered species, which is where Rich and I work, aquatic invertebrates, butterfly conservation, and pesticides. We're a science-based organization. We do research, we collect evidence, and use that to make decisions about what to do next about insect or invertebrate conservation. We work with researchers in academia, in government, all sorts of other contexts, and importantly, with community scientists like you all. At Xerces, we have a number of different really exciting community science projects, a few of which are listed on the left here. The Bumble Bee Atlas is a really prominent part of our work at Xerces, but we have these other cool projects. For example, a monarch milkweed mapper, where you can tell us where you see milkweeds around the West, and that helps us understand monarch habitat. We have a firefly atlas. We do freshwater mussel work, lots of cool stuff. So there's a link there if you are interested in getting involved in this science that we do. When the science warrants it, we also do policy and advocacy. This is our executive director. I think he's speaking in a government setting there. And we do a lot of education and training, such as what we're doing right now. We've worked in all 50 states and also around the world, training people about invertebrate conservation. And we also do a lot of land conservation, not the purchasing of land to conserve it, but to helping people to restore land through technical assistance and providing native plants and information about pollinators. So it's a 501c3 nonprofit organization, and we're sustained in part by membership. If you're a member, thank you so much for supporting this work. It's really important, at least I think it is. And if you want to become a member, there's a link there that you would follow to do that. So let's get started. Again, there are 3 modules. The first one is just about 20, 25 minutes. The second one is quite a bit longer, all of the methods for the project. We will then take a 5-minute break, just a quick little break, and then we'll get started with module 3, and we'll try to finish this all by 7 p.m. I will try to remember to tell you the bumblebees that you're seeing. And so in this image, this is 2 Bombus bifarius individuals, a queen and a worker, sitting on flowers in the Sierra Nevada. Okay, so why invertebrates? Why are we even here today talking about bumblebees? What's so important about invertebrates? It's been said that the fate of the world's insects is inseparable from our own, and I think that's true about other invertebrates as well. So we highlight a couple of the important functions that invertebrates perform in ecosystems here, including decomposition of organic materials, biocontrol in agricultural settings, as a food chain link, especially as food at the base of the chain for larger predators like this fish, and of course pollination, which is really the function that we're most interested in with respect to bees. And so pollination is this mutualism that bees and other pollinators have with plants. And the bees are there foraging for their food, while the plants need somebody to move pollen between male and female parts of the anthers, and that's pollination. So both parties benefit. That's why we call it a mutualism. This is really important to plants in general. About 85% of them need a pollinator, an animal, usually an insect, for pollination. To fully maximize their reproduction. And they're also important to agriculture. So about 10% of the total value of our food product is due to pollination. That's both managed bees and wild bees. And surprisingly, it turns out that wild bees are doing the majority of the pollination on farms around the world, whether or not honeybees are present. And that's an average in a meta-analysis paper that looked at many, many different studies. But that's an important result. It doesn't mean that honeybees are not important, but it does mean that these wild animals that we sometimes overlook are very important to actually producing the food that humans eat. So the major groups of pollinators, bees are not the only ones. There's a wide array of things that are actually pollinators, that are flower feeders. But moths and butterflies, flies, beetles, and wasps are super important invertebrate pollinators. And of course, bees. Bees are functionally much more important than these other groups as pollinators. And that is probably because they collect pollen for the food to raise their own offspring. I don't want to diminish the importance of these other types of invertebrates as pollinators, but bees are very important because of their dependence on pollen. So as pollinators, they have some interesting adaptations. The diet for these bees is nectar and pollen. So they have all sorts of adaptations for harvesting those two things from flowers. A long tongue that's like a straw, pollen harvest and processing and carrying apparatus. And we've circled here the pollen basket on the hind leg, this shiny hairless area. This is where the bee packs the pollen moistened with nectar for the ride home from the flowers. And so that's an adaptation for carrying pollen that only female bumblebees have. Male bumblebees do not. And it's important to understand that pollination is a mutualism, but it's not intentional. It's not altruism. It's incidental to bees foraging for their food, and the plants advertising the resources that are at the flowers. Before we go further, I'd like to just talk about the bee that everybody is aware of, regardless of how often you go outside or anything else, because they're culturally important, honeybees. And so in California, we see honeybees all the time. We see honeybees just about any place in the state we go. I think they're ubiquitous outside of the high peaks of the Sierras. They are a farm animal from Europe, Asia, and Africa that was brought here as an important farm animal, both for the production of hive products like honey and wax, but also for pollination. Honeybees are currently very important as pollinators of agricultural crops, and maybe a little bit less important as the producers of those wonderful hive products. But the pollination of agricultural crops is super important, and honeybees do that. However, honeybees are also widely distributed in the environment. They're feral or invasive, if you will. In California, just about anywhere you can find honeybee hives living without the aid of people. It turns out that honeybees have some negative impacts on bumblebees and other native bees. They compete with them for resources and change the behavior of those bumblebees in some cases. And they also spread diseases to them straight from agriculture. So honeybees are important to our food supply and are important and interesting in lots of ways, but they're not native insects here. They're not, they are wild, but they're invasive species like an invasive plant might be. So we need to think about that as a context for the story of these wild bees that we're interested in. So bees, how many are there? What types? There's a great diversity of these animals around the world. More than 20,000 have been described by science, and we know there are more. We keep seeing new species get described, and we're all in the field, we're all aware of like undescribed species that somebody found, et cetera. We know that in North America, there's more than 5,000 species of bees. And I think the figure for the continental US is about 3,600. That figure might not be quite right there. In California, we have 1,600 species. So this is a really highly diverse group of animals where we live, just thousands of different types. And they come in all sorts of sizes and colors and degrees of hairiness and life history. So these photos just show you a very few of the types of morphologies that we see in wild bees around the world and in California. Bumblebees are actually not so diverse. There's only 265 or so species globally, and in California, just 25 of those. So bumblebees, why do we study them? Well, there's a number of reasons, but one reason is just that they're these charismatic animals. They're big and furry and beautiful and really approachable. So there are other bees that need our attention, but bumblebees are a place that people often start when they're studying bees. So more about how bumblebees live and where they live. This is a plot that shows you the number of species of bumblebees in each area that is a hexagon there. And this is just as revealed by science. So this is based on a large data set of bee records, mainly from museum specimen data. So people digitized the museum specimen label and did that literally a million times or so. And so we have a lot of information about where bumblebees have been found in the past. So you see centers of diversity in the western Rocky Mountains, also on the East Coast there, and to some extent in California. Now, oh, and also the Black Hills is this very special little spot that has a lot of Eastern species and a lot of Western species. However, survey effort matters a lot to what we think the diversity of the bees is. Oops. Oops. I'm sorry, we are missing an image here. I'm sure, sorry. The image that I'm showing you right now is just the number of observations. There's supposed to be another image that drops in to show you the number of species in each of those places. And it seems to have been dropped by the slide deck. In any case, what I want to tell you here is that survey effort has a lot to do with how many species we think occur in different areas. And so there is some bias to this data set around the continent. Sorry about that slide. So how do bumblebees live? They are social insects and they have an annual colony life cycle. So a colony is founded in springtime, and by late summer or fall, it kind of falls apart and most of the bees will die. The only bees that will eventually survive are the newly produced queens that go out and mate and eventually hibernate. Um, they have a lot of communication as social insects. So there's a lot of communication of various sorts, pheromone-based, behavioral, and tactile. And they work in the dark in the nest. There's just a lot going on down there where they're coordinating the labor inside of the nest. There is a caste differentiation. There's different roles in the colony. So there's usually one breeding female, the mom, the large bee in the center photo, right in the center of the photo. And then her daughters are these worker caste individual females who do not normally lay eggs or breed. And then males are the other caste. Interestingly, there are bumblebees that are social parasites that attack other bumblebee species. And instead of going and foraging for their own pollen and making a nest, they just attack their relatives and force them to rear their offspring. In terms of food, they are dietary generalists. They can eat many, many different types of nectar and pollen, but that's the only thing they eat, nectar and pollen. There's rarely some other stuff they'll get into, but they're dependent on plants, but they can use hundreds, thousands of different types of plants. So they do have some flexibility that some other types of bees do not have. The nests are usually underground, usually in rodent burrows, but in a diversity of other types of situations, and some of them like to nest above ground. And we often see that species in birdhouses in California. All right. Oh man. I am sorry. This slide got messed up. Uh, this is a slide where I am meaning to show you the phenology, the timing of life history events across the season. And then the two graphs that you're seeing are supposed to come in after that, showing the actual data for phenology for these bees. So I'm just going to briefly describe this. I'm sorry about the slide quality here. I think I had a problem with the slide deck about 5 minutes before we started. Anyway, this is data from 3 years of the project for two species, for Crotch's bumblebee and Voznesensky bumblebee. And these are kernel density plots, which you can just imagine like a histogram. They're just sort of where the lines are higher, that means that we've collected more of that species of bee at that time. And you can see time is the x-axis or horizontal axis. And what I was trying to show with these plots is that there's a lot of variation in the timing of bees' appearance and the development of the colonies and the bees' commonness on the landscape and in the net as the season progresses, and a lot of variation between years. So just briefly, what you're supposed to have seen behind these graphs, in springtime, the queens emerge from hibernation. They go out and find a place to nest. They find food. They provision the nest site with pollen and nectar, which they then lay some eggs on top of. It's usually fewer than 10 eggs. They incubate those eggs, leaving only to forage. And it takes about 6 weeks before these first adult offspring emerge. And those offspring are female worker caste individuals who now take over all of the labor of the colony. And the queen now stays home and does nothing but lay eggs and eat food. And so later in the summer, as the colony grows, it's entirely female, a mother and many daughters. They decide to switch over to producing reproductive offspring. So now the females that they produce will be destined to become new queens, and the males that they produce are the reproductive males. After finding each other and mating, the colonies die, the males die. The only bees that survive are these newly mated, newly produced gines or queens that go out, find a hibernating spot, and spend between 2 and 9 months underground, depending on where on Earth they are, before coming out and beginning another nest. All right. Okay. There's one other thing about the life cycle that I wanted to briefly talk about. So on the left, we see an annual colony cycle diagram of a different sort for the bumblebees, and it's analogous to what I was trying to show you there. There's this other thing that happens with one lineage of bumblebees who we call the cuckoo bumblebees. They are bumblebees in every way. However, they are not social, they are parasites. They're social parasites who sneak into the nest of a host bumblebee species. They attack the resident workers and queen, usually with pheromones, but also aggression. And they kill all of the larvae, eggs, and pupa of the nest. And then they lay their own eggs and they force that colony to raise their own offspring. Really interesting behavior. The photo on the bottom right is not from California, but it is from a really cool study where this guy documented a cuckoo bumblebee trying to break into a nest box that he had crafted for the host bumblebee species there. They're also called Xerces with a P, and you may hear one of us use that term, but I'll try to say cuckoo bumblebee. This is our most common type in California, Bombus insularis, or indiscriminate bumblebee. So wrapping this module up, we actually have a lot of uncertainty about bumblebees, despite the fact that they are after honeybees, they're the bee that we know the most about around the world. They've been studied literally for centuries by scientists. Despite a lot of bees declining, we don't necessarily know exactly what's causing that. Some species are becoming more common than ever, and we don't really know why. We have some questions about habitat requirements and host plant use that are below the level of just telling you they're generalists. There's some stuff about nesting biology and the biology of finding mates that we don't know much about. And also this myopia about males. We generally study female bumblebees, and there are very good reasons for that. But males, the ecology of being a male bumblebee is sometimes overlooked. So for example, if you were to ask me about sperm and do pesticides affect sperm competence or something, there's very little information on that, and we should know more. So anyway, uncertainties that need to be unraveled and won't be unraveled by us in this project, but you can see that there is more to learn. Let's talk a little bit about the reasons to do the Bumblebee Atlas. And the big ones are really that there are declines taking place. So about a quarter of all bumblebee species are declining. And this is a quick roundup of the threats: habitat loss, pesticide exposure, diseases and pathogens, usually associated with agriculture, climate change, and most importantly, these interacting effects between these guys. I guess I also talked about honeybee competition previously. So all of these things interact to cause problems for some native bumblebees. It's not just one thing, and it's a death by a thousand cuts kind of a thing. What can we do about it? Well, you can think about ways that we can reverse some of those threats. In fact, we actually know a lot about recovering bumblebee populations. We know a lot about habitat creation and what is going wrong. And if we had all the sort of political will, we could probably do that. But one thing that we need more of, besides just like addressing the pesticide issue, is baseline data about where these animals occur. And so we do have a lot of historic information that comes from specimens in museum drawers, as I described, but we need more information about now. And so this is an image of our friend and colleague and mentor, Robin Thorpe, who was a bombologist in California at UC Davis, who did a lot of foundational work about the bees that we study now. And for Rich and I both taught us a lot about these bees. And the image of him is important because he actually established some of the baselines that we depend on when thinking about things like Franklin's bumblebee. So the California Bumble Bee Atlas is a community science project designed to train amateur naturalists or just the general public and get them out there collecting information about bumblebees. And this slide is designed to show that it actually does work in the sense that we cover a lot of ground and we reveal pattern in where species occur, such as the Western bumblebee in these green dots, mostly in the Pacific Northwest Bumblebee Atlas. The idea is here that community scientists were able to document that this bee no longer occurs on the Pacific Coast in Oregon, for example, where it used to occur. I want to just stop and say that community science is sometimes talked about by scientists as well as the general public as being somehow less rigorous or less serious than real science. And it is actually not true here whatsoever. This is the single best way to collect the data that we need to make conservation and management decisions about bumblebees. Rich and I, if we were given a giant pile of money and asked, how do you best want to solve this problem of creating a baseline for bumblebees? Personally, my answer would be, I want to do exactly what I'm doing now, because the California Bumblebee Atlas volunteers get it done. And the data is just as rigorous as what I could collect. So this really works, and you're contributing to what is actually the single best way to collect this data that we need to take action about protecting bumblebees. So this is the end of this first quick module, and Rich will now get into talking about methods after we take a break for questions. There's a link here to a quiz. You don't have to do it now. You shouldn't do it now. But at the end of this 2-hour period, you'll take this quick quiz. And if you pass it, when you pass it, you will be added to the scientific collecting permit, and you're ready to go with the project. You'll see that link again, but I just want to put it up there now that there's content in this presentation tonight that you'll be quizzed on. And that is just a thing that we need to do for the scientific collecting permit. So thank you. All right. And stop sharing.
Hey, Leaf, there's a couple of questions. First question was, since bumblebees are social insects, when I see one bumblebee, should I look for others?
That's a good question. They are social, and so they live in family units with lots of individuals. When we see one worker caste bumblebee out on the flowers, we should think of her not as a reproductive individual as you might think of a songbird that you see nesting or something. Because they live in these societies, most of the bees are not reproductive, and so a reproductive unit is the mother and father of that colony, not all of those other bees. So we do know if we see one bee, there's probably other bumblebees around, but that doesn't mean that they all go and forage in the same place. So we don't really know if we're seeing sisters. If we find one, are we going to find one of her sisters on the adjacent plant?
I also just noted that when I typed an answer to that question, and we found with some research that even in one location, if you find a bunch of individuals of the same bee species, it sometimes takes up to 8 or 10 captures of those to find individuals that are actually sisters from the same nest. So that's some evidence, I think, at least, points that bumblebees are probably incentivized not to forage directly adjacent to their nest. They're likely flying some distance away that's far enough so that they're not giving away the location of their nest to any particular potential predator, but they're also not so far away that they're wasting resources in the flight period. So anyway, that's how I read the situation.
Yeah, I agree.
Another question, do queens leave their nest to forage after her worker daughters emerge and begin foraging?
Normally, no. The queen does not leave the nest after the daughters start foraging. There's probably some situations where they would. For example, if the workforce dies or is reduced to 1 or 2 bees, then she probably would start foraging again. Springtime and colony founding time is a very dodgy time for bumblebees, and colonies can easily die with a week of bad rainy weather or some, sort of random event. When the colony is very small, there aren't very many individuals, and every one of them is very valuable to the enterprise.
I'll just note a lot of the questions that are coming up here are about ecology, and which is great. We don't have a whole lot of time to dig deep into a bunch of ecology tonight, but I did drop a link in the chat there. We are doing a nationwide ecology and conservation webinar that'll be dedicated like the whole 90 minutes all to ecology and conservation. So I would encourage you, if you are interested in taking a deeper dive, to join us for that. The link to sign up is in the chat. Maybe what time for, I don't know really how much time we have here, Leaf. Maybe one more question. Does that sound good? And then we should move on, maybe. Okay.
No, do one.
This is a quick one. How many individuals are in a colony?
That depends on the time of summer and the species. In the beginning, just one, right? Some species like Vosnesensky bumblebee, our most common and widespread species, have been documented to have colony sizes over 1,000 individuals. There's a wonderful little natural history study that I think Robin Thorpe was a co-author on from the Bay Area 45 years ago or something, where they dug up this nest, just an incredibly large nest. But typically it's much smaller. It's in the low hundreds. Some species, it's not even 100 bees before the end. So it varies quite a bit. And those those are distinct life history strategies. The get huge before you reproduce is a distinctly different strategy than create a nest of 35 individuals and then start reproducing. So it depends on ecology and probably their evolutionary history as species.
Yeah.
Yeah.
And even within a meadow, you could probably find differences between species, right? You'd find one that had 1,000 because they got
Yeah.
everything right, and one that maybe had 200 because they got
Yeah.
started late or something happened, it just varies. Yeah.
Yeah, actually, you bring up an important point. The one I said is the most common and widespread. Vosnesensky bumblebee has huge colony size. So by the middle of the summer, you'd almost expect to see more of those bees than one of the species with real small colonies, just because one colony can send out hundreds of individuals. And so this brings up an interesting thing about being a bumblebee scientist. You're never sure, how many colonies are sending individuals, how abundant the bees are in an area because they're social, right? And so, yeah, it depends on the species. All right, well, let's turn it over to Rich for the methods, and I will be answering questions and dropping links in the chat.
Great. Leaf, can you see my title slide here?
Yes, I can. Looks good.
Okay, great. All right. I just also want to note, I just put a link in the chat. Someone asked where we post the videos. They are on the Xerces Society YouTube channel. We also typically post them on the Bumblebee Atlas website. It is a little confusing for some of you because there's currently a California Bumblebee Atlas specific web page, but there's also like our nationwide project, Bumblebee Atlas, and Leaf is in the process of migrating the California Bumblebee Atlas website over to the Bumblebee Atlas website. So fairly soon, everything is going to be in that one-stop shop Bumblebee Atlas program. We'll set up redirects so that if you have a link saved, it should take you at least to the Bumblebee Atlas page. So if that link is a new one for you, if you're used to going to cabumblebeeatlas.org, bumblebeeatlas.org is going to be the new site for you to come to. So just wanted to, if you got confused, that's the source of confusion, and we're sorry about the confusion, but as we've grown, we've got growing pains and we're trying to clean house. So there you go. All right, I'm gonna leave sort of giving you a bit of understanding of bumblebees and a little bit of justification for why we're running these projects. I'm gonna try to get into the nuts and bolts of how we actually do this or how you will actually do this. I will just say that this is not really the best thing to fully learn on YouTube or in a webinar. If you've never, captured bumblebees before or taken pictures of them or participated in a project like this, I would really strongly encourage you to visit the events page and sign up for a field event and go meet Leaf in the field and spend a couple hours learning from one of the best about how to do this. It's a really a fabulous opportunity. You'll get to interact with other volunteers too, and just have a good time and help build communities. So I'm certainly going to share some information for you about how to do this, but I would encourage you, especially if you're new to this, to try to seek out a live event and attend in person. So here is the outline. We've been through the introduction, the ecology, and we're now getting into sort of the A to Z. As Leaf mentioned, we are running these programs in 20 states from coast to coast. We have a whole host of partners in this, from state wildlife agencies to federal wildlife agencies and federal the Forest Service and the BLM are both engaged in this project in different states, and we have a whole bunch of other nonprofits that are engaged as well. So it's a big collaborative conservation project. So while you're participating in California, you're really contributing to a much larger effort that we can talk about this and we have in other places, but that is really driving real conservation results on the ground and impacting how we're protecting these species in real meaningful ways. So everything that you do for this project is truly making a difference. You're contributing, as Leaf said, as much as any professional scientist would in ways that we couldn't. As much as I would love to take a lot of money and go survey the bumblebees of California, it would take Leaf and I several decades to do that. We'd have a great time. But by the time we were done, the information that we got at the start would be obsolete. So this is really the only way we can do this project. And thanks for taking the time to learn how to do it. Okay. Just quickly, the progress to date. This project started in 2022 under the purview of Leaf, who's been leading this project since day one, very competently and wonderfully. You're all lucky to have him. As of the end of last field season, we had around 1,200 or close to 1,200 permitted volunteers that have contributed almost 20,000 observations of bumblebees, and nearly 2,000 point surveys, and surveyed around 60% of the state. So tremendous progress towards really completing the atlas. As you can see, if you look at the map on the right here, there are still some gaps, and that's really our focus for 2025 is how do we fill in that map and close those gaps and get to some of the areas that are in the middle of the Central Valley or down in the Mojave. How do we fill in some of those gaps and get some surveys done? We still want to get surveys where we're completing them as well, because long-term data sets, as you've learned about Leaf's conversation with Robin Thorpe, are so important, but also getting outside of population centers, getting outside of transportation corridors into some of the remote wild areas of our state is really where we're going to learn the most information about how species are doing and potentially why. There's more at the California Bumble Bee Atlas specific website. There's a highlights page where you can learn more and get into some of the more detail of the results. So if you're brand new to the project, the best thing to do is just go to californiabumblebeeatlas.org, and you can find our Adopt a Grid Cell page. You can see that we've put a new priority grid map for 2025, as you can see on the map on the right here. And the high priority grid cells are grid cells that have had 0 surveys in them, I think. Although it's sort of a mix, I guess, of areas that haven't been surveyed versus where we think we're going to find high quality bumblebee information. So the green cells are really where we're lacking survey information, and we think we think there's a lot left to learn there. The blue are sort of our medium priority grid cells, areas that would be great to get another survey in or to do a single survey in. Some of those grid cells in the Mojave, we're probably going to end up with a lot of zeros down there, but those zeros are just as important as surveys that document 100 bumblebees, because otherwise we have to make assumptions about what's there and what's not there, and we won't know unless someone goes to look. So filling in these gaps is really our goal for 2025. The best thing that you can do is adopt one of these grid cells, one of these high priority or medium priority grid cells, and then follow the standardized protocols that I'm about to lay out and submit those data to Bumblebee Watch. The ideal is if you adopt a grid cell, the ideal is that you are committing to conducting 2 surveys in that grid cell this year. I will say that if you feel like you can't commit to 2, but you can do 1, don't let that stop you. There's a lot of people in California, a lot of people interested in this project, a lot of volunteers that are engaged. And, if you feel like that barrier of 2 surveys is too much, I would say sign up, go do 1. More than likely, you're going to get the bug and want to go back and do it again. But if you don't, it's okay. Like 1 survey is very helpful and it will contribute to the project. So don't let this barrier of 2 as a minimum stop you from signing up from the project and going out and conducting a survey. We really want every survey that you can do, no matter where you do it, is helpful for the project. And I'm going to talk about these other 2 steps of following the standardized protocols as we move through this slide deck. So catching bumblebees is something that's not easy to do. I will say on that link that I shared right before I took over from Leaf, I shared like the Bumblebee Atlas training, online training website or web page, I guess I should say. And on that, one of the videos in there shows a video of me at the beginning of COVID when we were trying to figure out how to adapt this program when we couldn't be in person with people anymore. Or yeah, I made a video and I show how to catch bees like in more of a live action way rather than just showing pictures of people doing it. I would encourage you to go watch that video. I think it's still an effective way to sort of at least see some visuals about how the movement goes and what the different options are for trying to get a bee into a net and then a bee from the net into a vial. Those are probably the most complicated parts of the project, I think. And so, this is something I would encourage you to practice at home, like in your backyard, at a local park, before you go out and start saying you're going to conduct a survey. Because ideally, everybody that participates in this project, we want them to have relatively equal skills. And that's impossible, right? Leaf and I have swung a net, I don't know how many times, many, 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 many times. Oh, for the last 30 years, right? Both of us have been doing this for a long time. This might be your first time, and there's no way that you and I could ever have equal effort when we're doing a survey. But ideally, like everybody at least has some minimum competency. And so you've been out there, you've tried this, you know that you can catch a bee and transfer that bee successfully into a vial and get it into a cooler. And so I would encourage you to just practice. The bees are pretty hardy. You're not going to harm them. And it's a pretty, it's relatively fun as well. If you have kids love to do this. So it's a fun thing to just go out and test out in your backyard and do a little trial. But, like generally speaking, as long as you're dealing with relatively abundant flowers, you can swing relatively hard through a flower to capture the bee into the net. You can also put the net down sort of over the top, over the vegetation, and then that'll allow the bee to sort of fly upwards. As you can see in that photo here in the upper right, you can see the individual is holding the net over the flower and then lifting the net up in the air. And bees tend to fly towards the light, so they'll go up in the air, and then you can sort of grab the net below the bee, and then you'll have it captured in the net. And yeah, you just have to practice this. You have to go out and try what works for you. Everybody has a different method that they like. Sometimes different plants have different things that work for them. Sometimes there's some thorny plants out there that if you tried to swing a net through it, it isn't going to work and you're going to shred your net. And so you have to try different things in different places and just become familiar with what works for you. Once you have the bee into the net, it's really important to sort of turn the net over once or twice to sort of close off the top of the net. Otherwise, the bee is just going to fly out of there as quickly as they possibly can. So you want to try to sort of turn the net over, and then you'll at least have the net sort of serving as the top of the net hoop, and then the bee won't be able to fly out. And then you've got to work to try to get the bee into the vial. And again, this is something that's hard to talk about. It's much better to show you how to do this. So I would encourage you to attend a field event. But you're basically going to stick the jar or the vial that you have into the net and sort of pin the bee in the open space against the net, and then either put the top to the vial on in there or drag it towards the edge and then put the top on. It's just something you have to look out for. If you're going to get stung in this whole process, it's likely going to be in the transferring process, because the bees aren't happy. They don't want to be in a net. And you don't want to have them in a net, and so they're stressed out, and you're stressed out probably, and they're making noise, and I would just encourage you to just like take a breath, slow down. There's no rush here. Just take the time to figure it out, get the bee in there, make sure everybody's safe and happy and healthy, and move on. So just, yeah, again, this is something that's worth practicing a fair bit before you get out into the field to conduct a survey. I will say the other thing is once you've captured the bee into the vial, typically what I recommend is we also want to know what flower that the bee was visiting. So it's really important that you document that flower species. And there are a few ways to do this. What I like to do is I like to put a number on the top of all of my vials. And then I take a picture of the number on the top of the vial next to the flower that I captured the bee on. And then I have that photo later when I'm processing my data. You can also take a little bit of the flour and put it inside the vial with the bee, or you could put a note on the vial with a grease pencil or some other option like that, or with a Sharpie or whatever. So just make sure you think about that. Is we also want to document the flowers that we're catching these bees on, and I'll talk more about that process later. But since I'm just talking about capturing bees here, I wanted to at least to mention it. And again, just another plug for the events. There's the URL there on the page. I'm sure Leaf can drop it in the chat for you as well, since it's not clickable when it's on the screen there, but it would be in the chat. Just want to make sure, like, your personal safety is more important than any surveys that you could ever do for us. So please make sure you're taking care of yourself out there and anybody that's out with you. There are hazards out in the world in California. There are rattlesnakes. There's poison oak. There's lots of other things that you could get in trouble with. And in some of these places, there's no cell service, and so you're kind of stuck back in the 1990s. And so practice and think about your field work as if it were the 1990s, and bring a paper map and make sure that people know where you're going to be, and make sure you have a backup plan. Make sure that you have permission to be where you're going. We've put some resources together to help you with this. There's, Leaf has put together a whole host of permits that we have for a bunch of places. So please reach out to him, or maybe there's a spot on the website where some of that information is outlined. We've tried to put lanyards together for people in the past. I think we'll be doing that again this year, for a little bit of field safety. There's a placard you can put on the dashboard of your car to let folks know what you're doing and why you're there. And if there's anything else that you need to feel safe in the field, please don't hesitate to reach out to us. We're here to help you do this project. And if there's something we haven't thought about or something that would be helpful for you, please reach out. I'd also encourage you, this is fun to do with friends, with other people. I mean, it's fun to do by yourself too, but you're safer in groups. And if you're, interested in doing this, but it sounds scary or you don't want to go out alone, I would encourage you to go to the private Facebook group that Leaf has set up, and just sort of say, hey, use it as a matchmaker, and say, hey, I live in Santa Rosa and I'm interested in doing surveys. Is there anybody nearby that wants to meet up and do a survey? And you might make a connection with somebody and maybe make a new friend. So anyway, just an idea, a thought. It is more fun with people, with friends. The way that once you've captured the bee into a vial, the way that we are able to document them, what species it is by taking detailed photographs. And we do that by initiating cold anesthesis, or using cold anesthesia for the bees. And so we essentially put them to sleep by putting them at a cold temperature. And that cold temperature ideally is right around freezing, right around 32 degrees Fahrenheit, but it shouldn't be significantly below that. And so just want you to be aware, if you have like a chest freezer that freezes ice or whatever is in your freezer well below freezing down to 0 degrees Fahrenheit, that can be really dangerous for the bees and can actually cause mortality. Likewise, some of the newer blue ice that are on the market, like the Yeti and other sort of premium brand names that are out there, some of those are also formulated so that they freeze down to a very low temperature that's too low for bumblebees. So we just want to make sure that we're not using those. Or if you do have a chest freezer that freezes down to 0, just leave the ice in your cooler for a while before you start to put bees in there. Maybe leave the lid open for a while and let the whole cooler raise up to more around freezing. You can use those blue ice things at the bottom of your cooler to sort of keep the ice cold, but bees should never go in direct contact with those blue ice things. That can be dangerous for them. So please be careful. We don't want to hurt the bees that we're trying to document. We're trying to conserve them after all. So that's why we've developed this non-lethal technique, and we want to make sure the bees are safe. So we've found that for the most part, at least I haven't heard of many or any documented cases of sort of gas station ice going to, causing any harm. That typically seems to be about the right temperature. Likewise, if you're traveling and if you're in a hotel ice is the right size and shape to put I found it to be like the best option for sliding vials in and out of, and the temperature is perfect. So feel free to use that as a resource if you're traveling and to do surveys. As far as when to look for bees, that answer is going to vary quite a bit just because California is so large and diverse. There are so many different habitats from the coast to the high Sierra. And really pay attention to the conditions. There are different opening seasons, I would say, that Leaf has established on the website. So go take a look at the map as to when each season starts. I believe that map is the top of the Adopt a Grid Cell page. So take a look at that. Excuse me. But roughly, the general guidings in California is for Southern California, the season is roughly March to September. For Central California, and the Coast Ranges and the Central Valley, it's April through September, and then for Northern California and in the mountains, May through September. I will say that May is probably early for the mountains. I would wait till June, maybe even mid or late June, based on my experience working in those areas. But the season's open. So that's when to look for bees, sort of as a project stance. Like if you're doing a California Bumble Bee Atlas survey, we'd like you to do it in these windows in these places. Of course, bumblebees are active earlier than this. There are bumblebees active probably year round in many of the places that many of you live, but it's only a couple of species. And the whole point of this project is for us to document the species richness across the state. So if we do a survey at a time of year when only one or two species are active, then we're unlikely to detect, or we're not going to detect all of the other species that may be there. And if only one or two surveys are done in a grid cell, then we've missed a whole bunch of species and have incorrect information about the species that are living there. So that's why we're targeting these sort of windows. We're trying to hit the peak of when the most species are active in most places. And so this is general guidance. But if you get to an area and you see only a few flowers and you only see queens flying around, it's probably best to not do a survey, to be honest with you, and to come back in a couple of weeks when the phenology has changed a little bit. So use these as a general guide, but you can also use common sense. And if you feel like you're not sort of knowledgeable enough to have that common sense, don't hesitate. Do that survey, submit the data. It's still great and it's still helpful. It'll help us learn a lot. I'm not trying to shy you away or give you too many choices to make, but I think folks generally know when things look like it should be hopping. And if it's not, come back. In terms of where to look for bees, there's a whole host of options. Like your own backyard, of course, or land that you own and manage is great. Private lands where you have permission to access are great. Roadsides generally speaking, have a right of way along the sides of them, allowing you to access it, no matter who the surrounding landowner is. So, be careful and make sure you're aware of what those rights of way laws are in the area that you're surveying. But generally speaking, those are accessible to you. And then there's a whole host of public lands, national parks, national monuments, California state parks. All of these different public entities, especially in California, have regulations about what you can do there. Some of them require permits, some of them require letters, some of them require a whole lot more than others. So if you have questions about this, please reach out to Leaf. He can help you get the permits and access that you need. In areas like the Forest Service and the BLM, we just sort of have general blanket letters that we can send you, but you need to have with you when you're in the field that says that you have permission to survey there. Those are relatively easy. Some of our national parks, require you to be a named individual on the permit, and you have to let the land manager know that you're going to be there and when and where, and to coordinate with them. So please reach out to us if you're going to be going to, especially any sort of area that's managed for biodiversity specifically, like a national park or a national monument, or any area that sort of, has a higher sort of standard for visiting it, like just keep that in mind. And we can help you get those permits, or let you know whether it just may not be possible in the time frame that we have. But please don't go somewhere unless you're sure that you have permission to be there. We don't want to have access revoked from us because we're sending people all over the state where they don't have permission to be. Anywhere that has flowers is a great place to look for bees. Generally, most people think about meadows or the rolling golden hills of California when they're blooming. But there's also forests. Oftentimes have forests or have flowers in them that bumblebees are visiting. So, don't hesitate to look for places to survey in areas that are outside of what we sort of think of as stereotypical bumblebee habitat. So anywhere you find abundant flowers, you're likely to find bees, and we're interested in finding what bees are using which habitat. So getting surveys in diverse areas is really important. Yeah, we're using a non-lethal protocol to document the bee species that we find as part of this project, and I'm going to teach you more about how to do that. So your charge is really to go out and just find bees on flowers and capture them and get them in a jar, and then we're going to do the identification for you, which is really nice. Your job is to sort of catch and photograph, and our job is to do the technical identification piece of things. So when you go out to do a survey, it's really like you want to be flexible. So you can do a bunch of planning at home, but when you get to the field site, what it looks like is may not be what it looked like on Google Maps or in your atlas. It may change. So just be flexible and try to move around a little bit and find a place that looks like it's going to have a lot of bees in it. That's generally what we're trying to do. It doesn't need to be just like a random point and you're like, oh, I selected this, so I'm going to do a survey here, even if I don't find bees. What we're really trying to do is find out what bees are visiting what plants and in what habitats in the state of California. And so we want to find them. And so it's okay if you sort of look for the areas that look like they're going to be good for bees. That's what we're sort of hoping that you're going to do, as long as we're doing that across a variety of different habitat types. For point surveys, so there's 3 different types of surveys. Well, really just 2. There's roadside surveys and there's point surveys. And for point surveys, we want you to find a patch that's about a hectare. And a hectare is about the size of a soccer field. And then if you're doing a roadside survey, you're actually going to find 5 spots along a roadside that will make up one roadside survey. And at each of those spots, you want to find excuse me. You want to find a large patch of flowers such that could maintain your attention for like 10 to 15 minutes. That's sort of the idea that you would want. So, not just a few small flowers, but like a patch that you could walk along a roadside at. And then you really want to make sure to look on every species of flower. Humans tend to be attracted to like really showy flowers. And we go to those, the dandelion, the big yellow thing that sticks out, the green, bluebells that are showing here. Or I'm sorry, whatever. I'm sorry, I'm misspeaking, I think. But you know what I'm saying. We tend to go to these big showy flowers, but remember that bumblebees see a different part of the visual spectrum that we do, and they're cueing in on different things that we are. And most important to them is where's the pollen and where's the nectar. And so, there may be a non-showy brown or green flower sort of underneath the understory of what you're looking at, that bumblebees are visiting. And we want to be really careful that we're looking at all the flowers and not just the big showy flowers to find out what bumblebees are using. So because of this, we encourage you to use your ears as much as you use your eyes when you're out doing surveys. You'll often hear bumblebees before you see them, and make sure that you're paying attention to those cues. If you're out there surveying with somebody else, try not to have a conversation while you're surveying, because you want to use all of those cues that are available to you. And yeah, look for bumblebees on every species of flower that's in your plot. We're going to catch those bumblebees, put it in a net, put it in a vial, and then put the vial into a cooler And then we'll process those bees at the end of our survey period. For your surveys, or to go out and do surveys, this is a general list of equipment that we recommend. You will definitely need an insect net, and you will definitely need a cooler with ice inside of it, and you will definitely need vials. And then you will also need some way to document those or to take pictures of them. So this could be a an SLR, like nice camera. It could be a point and shoot camera, or it could be your smartphone that you already carry around in your pocket. All of those things can work. They will just all require you to practice again, and I'm going to talk about that more a little bit later. You'll also need the data sheets, which are available from download on the website, and then some form of timer, and then ideally maybe some maps and some field guides and a notebook and some other options on here just for you to consider. Drinking water is always important. Sunscreen and a hat. Make sure you're taking care of yourself while you're out there in the field. As I mentioned, there's two options for formal surveys. One is a point survey, the other is a roadside survey. I'm going to talk about each of these in turn here. A point survey is as I mentioned, is in about a hectare. A hectare is around the size of 2.5 football fields. It's about the size of the infield of a track if you were a track and field person, or about the size of a soccer field. A little bit bigger than that, but around that size. That's what we're thinking. You don't have to measure it. You don't have to mark corners. It's just approximately this size. And what we found is that if you have 45 minutes to wander around looking for bumblebees, you're probably going to cover around a hectare. And you do want you to try to cover that space. So make sure that you're wandering through the whole habitat while you're doing your survey, and you're not just focusing on the one plant that seems to have a bunch of bees on it, because there might be something else, in the far corner where there's different species of flowers and different species of bees. We want to make sure to capture all of that. For a roadside survey, as I mentioned, you're going to take a section of road that's 10 miles long, and you're going to do 5 surveys along that 10-mile stretch of road. Each individual survey should be separated by at least a half mile from the last site. We want to avoid the possibility of capturing the same bee twice. So that's what this distance is about. Each survey that you conduct, instead of being 45 minutes long, is going to be 15 minutes long, and you're going to do 5 of those, and that completes one roadside survey. And you're going to do a habitat assessment, which I'll talk about more in a little bit here, at each of those 5 sites. So again, I'm going to talk about each of these in a little bit more detail moving forward here. You can also do something called an incidental survey that isn't really a survey. It's more just like kind of an iNaturalist thing where you're out in the field, you're in your backyard, and you take a picture of a bee and you want to share that information with us. Great, please do it. It does not fulfill the requirement of sort of a survey, but it does help us understand what species are active when and where and what flowers they're using. But we also lose a bunch of information. So we're not sort of necessarily encouraging you to participate in the project by going out and doing incidentals. But if by participating in this project, you just start loving bumblebees and want to take pictures of them all the time, please share them with us. We're more than happy to help you identify them and to gather that information. All right, so jumping into a little bit more details about point surveys. Again, think about your personal safety, plan a few locations before you start traveling out there, and remain flexible as you go out there and make sure that somebody knows what your plans are. Once you have found a location, you want to, again, pick that hectare of space. And then your survey, each individual survey is going to be 45 person minutes long. And what we mean by a person minute is that if you are surveying with just yourself, you're going to set your timer for 45 minutes and you're going to conduct your survey for 45 minutes. Every time that you get a bee into the net, you're going to stop your timer, because it's not, again, to even the effort that I was talking about earlier. We don't want to calculate how long it takes you to transfer a bee into a vial from a net. We want to find out how many bees you can capture in 45 minutes. So the 45 minutes is just the search time. So you're going to pause the timer as soon as you capture the bee. And then once you get the bee into the vial and the vial transferred to the net into the cooler, then you can start your timer again, and then you'll go and find your next one. So that you're going to do that for 45 minutes, capturing every bee that you find. If you are working with another person or two other people, let's say, then we want a total of 45 person minutes. So that means that each of the three people is going to survey for 15 minutes, and you're going to spread out in your hectare, right? And each person's going to survey for 15 minutes. So in total, it's 45 person minutes, but each individual person has only surveyed for 15 minutes. So that's what we mean by person minutes. Take the total number, 45 minutes, divided by the number of people you have participating in your survey, and then each person does that number of minutes, which adds up to a total of 45 minutes. And again, we're going to pause each person's going to pause their timer when they catch a bee, or if they need to take notes, or if they need to blow their nose, or if they need to sneeze, or whatever. We want the 45 minutes to be truly searching and looking for bees. The best thing about point surveys is that you're there for 45 minutes. So you're getting a really clear picture of who's there. You're likely to encounter most of the species that are there on any given day, and we're going to get detailed information. Also, point surveys tend to take place in more remote and wild areas, and this is where we have native plants and native habitats, and they're generally intact. So we're getting a lot of really good information here. The problem with a point survey is it's only one spot in a 50 kilometer by 50 kilometer grid cell. So we're getting while it's a detailed about one small area, the information is diluted as far as the whole area is concerned. So you may miss species that aren't nesting nearby, but are nesting 5 miles down the road or whatever. But these are the preferred method. We prefer point surveys because of the pros that you see here. They give us the most bang for the buck, for lack of a better term. Roadside surveys, again, be flexible here. You can pick out a 10-mile section of road, and then when you get there, you might find that there was a fire or that there's nothing there. So remain flexible, and try to plan a couple of different areas. Be respectful of any signage that you see. Take care of yourself. Make sure that you're wearing bright colors if you're doing a roadside survey, that you park your vehicle safely, that you don't, just be careful, and respect all of the signs and regulations that you see out there. Once you get to your 10-mile section of road, you're going to start driving down the road until you see a patch of flowers that is along the roadside in areas that you can access. And you're going to do basically a point survey, but it's in a smaller area and it's only 15 minutes long. But you're going to follow the same procedure. Every bee that you capture, you're going to put into the vial, put into the cooler, document the flower, and then start your timer again and do that for 15 minutes. And you're going to do 5 of those along this 10-mile stretch of road, each individual site separated by at least a half mile. And then, when you submit your data, you're going to submit 5 different surveys, but you're going to name them similarly to each other so that we can associate all of that as a roadside survey. That's really the difference between a roadside survey and a point survey. Oh yeah, here's the note. So if you are doing a roadside survey, please, when you're naming each individual site, start with the same name and then a number after it, so that the first one on a roadside would be 1, 2, 3, 4, 5. Pretty straightforward, but please do that. It's really helpful for us. The best part about roadside surveys is that we're getting more individual survey locations. So our surveys are spread out over a larger area, which potentially gives us a chance to encounter more species that aren't all nesting in the same area. So we're going to get different habitats, potentially different species, and they're generally easier to access. The cons is that we tend to find a lot of invasive plants near roads, and the surveys are shorter. So we have a shorter amount of time to encounter all the species that may actually be living in that individual area. And for this reason, we do prefer those point surveys. And then your incidental observations, these are just, you see a bee on a flower, you want to snap a picture, go ahead and try to do that. They can happen anywhere, but it's really important to remember that bumblebees, as you'll learn from Leaf in a bit, are hard to identify. We still need high quality photos. A picture of a bee from 10 feet away, like on a flower where you can only see its butt sticking out of the flower, is not going to give us enough information to identify it as species. So please, try to think of yourself a little bit as a photographer and get those characteristics that we need to identify it. This photo here on the right is beautiful and it's great, but it's not enough, at least for me, to identify that bumblebee to species. There's too many lookalikes that it could be. I can narrow it down to a few species, but I can't get to species. So just as an example. But yeah, then you can just upload those photos to Bumblebee Watch. The good thing about them is they can happen anywhere. There's no real time commitment. You don't have to follow necessarily the conditions on the SCP here, because you're not capturing or handling the animals. You're just taking a picture of them on flowers. But the downsides are we have no measure of effort, which means we can't assess, did you look for all the species? Was another species there? We can't figure that information out, and we don't know what habitat's there. And also there are just the con here says there's more human bias, but there's also some bee bias here as well. There are some species of bees that just tend to have slower behavior and they're easier to photograph. And so I think there's also some bias from that perspective as well. It's really important that we take care of our bees. I can't remember if Leaf mentioned it or not, but one of the potential causes of declines in bumblebees is actually disease, and disease transfer. I think he mentioned it in relation to honeybees, but I'm not sure if he talked about, even within the bumblebee world, there are some diseases that are potentially have caused declines in the past and sure could in the future. And what we don't want to be is we don't want to be vectors for moving those diseases around. Bumblebees are pretty good at moving them by themselves, but we don't want to be those vectors. We don't want to be the people moving them around. So it's really important that between survey locations, at the same survey location, it's okay because bumblebees are moving diseases around better than we could at the site. But between survey locations, like if you're moving to go do a new survey, it's really important to sanitize all of your equipment. So your nets and your vials should all be sanitized. Warm soapy water works. Ethanol or alcohol wipes can work. A mild bleach solution. These are all options that work and can do it. It's just really important to do this in between each survey location. You can carry a mild spray bottle in your car that has, disinfectant in it, spray your net, wipe your net around in the air and dry it off. In California and most habitats, it's going to happen really quickly. And it's just an important step to take to make sure that we're keeping the bees safe and we're not the ones that are moving diseases around between sites. In addition to that, it's important that when you do have bees, like in a net, I'm sorry, not in a net, in a jar, you don't want to keep that jar outside of the cooler for very long. A couple of minutes is okay, but it's essentially can turn into a greenhouse very quickly, particularly if it's in the sun. And the temperature inside there can get too hot, beyond the thermal tolerance of bumblebees. So please make sure to get them in the cooler, in a relatively rapid time frame. And then we want to leave the bees in a cooler for no more than 2 hours. That's sort of the goal. That should give you plenty of time to process all of the bees from your survey. And then when you release them, make sure you're doing it relatively close to the location where they were captured. Within 100 meters is the guidance that we've been given for our scientific collecting permit. So please follow that. If you're planning on trying to do surveys, this is a very important slide. This information is covered in the quiz that you will see. So take a screenshot of it, write the information down. This is information that's important and you will see again. Once you've sort of completed your survey, whether it's a point survey or a roadside survey, you're going to have a bunch of bees in a cooler that it's time to document. When we do that, we want to make sure to take the best pictures that we possibly can. This is really your job as a volunteer. If I'm to be sort of the manager here, and the director of this project is to like, if you've gone through all this effort of all of you've taken this training, you've planned your survey, you've traveled to your survey site, you've spent 45 minutes, like looking for bumblebees and catching them, and you've put them in a jar and you've put them in a cooler, and like you've done all this amazing work. If at this point the pictures that we take aren't good enough to actually identify the bees that we had in our survey, then a lot of that effort has unfortunately maybe not been wasted, but not used to the best of its potential. So taking photographs is really an important aspect of this project. And like capturing a bee in your backyard, I would really encourage you to practice taking pictures before it really matters. Get used to using the equipment that you have. It's possible to do with a cell phone. It's possible to do with the most point and shoots, but it's going to take some figuring out how to do it close up, how to get a photo where you can see the bee. A good piece of advice here is if when you look at the photo, either on your phone or like on the little screen on your digital camera, if you can see individual hairs, like you can see on this bee here on the left, then that photo is high enough quality that we can probably see the features that we need, generally speaking. That's a sort of general guidance. So try to take pictures so that you can see individual hairs. Along with that, we want to take a number of different photographs. The compilation of photos that you share with us, each individual on Bumblebee Watch can get 6 photos. So you can take up to 6 photos and share them with us. So that's a lot of photos to get the pieces of information that we need. But we need to see the hind leg. The hind leg is going to tell us whether it's a male or a female, and whether it's a cuckoo bumblebee or a true bumblebee. We want to be able to see the head and the face and what color hair there is. And then ideally, we also want to see this space called the malar space that's below its eye and above its cheek, or I'm sorry, above its mandibles. So we want to see that a picture, so an angled picture of its face. I'll try to share a picture of it with you in a little bit here, just so you have a little bit better idea of what that is. And if I don't have one, I'll make sure that Leaf shares one with you when he goes over the ID portion of this. We also want to make sure that we see the color pattern on the thorax. The thorax is where the wings are attached and where the legs are attached. It's between the head and the abdomen. It's that middle part of the body. And we want to see the color pattern on the top, in front of the wings, in between the wings, and behind the wings. And we often need to see the color pattern below the wings as well. Let's see if I can use the laser pointer here and just show you what I mean. So this is the area in front of the wings that we want to take a look at. This would be the area between the wings, and then behind the wings is a little bit obscured by this wing here, but we want to be able to see that color pattern back there as well. This is the area below the wings that I'm talking about that we also sometimes want to see. And then we also want to see the color pattern on the abdomen. A picture from the top like this, where we can see every segment of the abdomen, is really important. So we can see the striping and banding pattern on each segment of the abdomen. It's not obscured by the wings. It's not out of focus. It's not too dark. It's perfect so that I can actually see where the division is between each of those segments and see what the color is on each of those segments. So again, this is another time where getting some practice by going to a field event it's a really good idea, where you can see how Leaf does this. He can train you how to use your camera, and show you, some techniques and tricks and that he's found with different devices over the years of doing this project. So if you have questions about how to do this, please attend an event or email Leaf and ask for advice. I have this kind of camera. What's some advice that you might have? So here's some just more detailed pictures walking through each of those individual photos. So again, I need a picture that shows me the head, where I have the front of the face and the color there. So I really want to see, just getting the laser point out here, I want to see the color pattern on the front of the face here. And I also want to see the top of the head here, what color pattern that is. This is not a good photograph of the cheek. I mentioned that cheek space, that malar space, which is right here basically on the bee. Ideally, we would be taking that photograph from over here. So you'd be looking at the bee this way, and you'd see directly that space below the eye and above the mandibles. And again, if there's a better photo of that later, I'll share it with you. This is the photo that shows the thorax really well. I can see in front of the wings, I can see between the wings and behind the wings, and also below the wings. And then also, then I can see the abdomen. And here, this is not a great picture of the abdomen because a lot of it is obscured by the wings. I can't see what's in the middle of T1 and T2, which are the tergal segments that I'm referring to here, are these different segments on the abdomen. And we really need to be able to see the entirety of those segments. So if you need to, feel free to pry those wings apart and take the photo from straight down on that abdomen so that we can see the entire segment. You can just stick a pencil in there and just gently pry them apart while the bee is chilled. And that should give you plenty of time, and the bee is plenty safe that you can actually do that and take a good picture. Or you can bring a pair of forceps out into the field with you and use those to peel the wings apart. Generally speaking, when you've chilled a bee and it's fully out and ready to take a picture of, you have about 5 minutes to work with. Sometimes more, sometimes less, depending on the ambient temperature and the humidity out. But you've got about 5, sometimes less. Maybe I should say 3 minutes in California. I don't know, Leaf. Maybe you can let us know in the chat there. But you do have a little bit of time, so you don't need to panic. You have some time to work with, and again, this is a good reason to practice at home. And just so you know a bee is ready to process when it's in the vial, it's been in the cooler, and you can take the vial, and if you just gently shake it back and forth, I don't mean like shake it, like this, just gently back and forth. If the bee is sort of not able to fight against you and they're sliding on the bottom of the vial, then they're ready to be processed. If they're not, if they're sort of walking around or moving, they're probably going to warm up too quickly, and you want to put them back in the cooler and let them chill down a little bit more. There's nothing wrong with putting a bee back into the cooler. That's a fine thing to do so that they continue to chill. And again, just a reminder, like, please try to take photos that are in focus and that have adequate lighting to see what we actually need to see. These are just some examples of, some people that have been out doing surveys that have submitted photos that, just just don't quite cut it. They don't allow us to identify bees. There's just some photos, even with good high quality photos, where we may not be able to identify the species. It may be really worn or a unique color pattern or whatever, and that happens. It's okay. We don't need to identify everyone, but we'd really like to at least have the photos that we'd like to see, or be able to see details so that we can, make a determination if we possibly can. And there's just a photo of like spreading those wings apart. So you can see if, those wings are obscured, I might miss that crescent of brown here on T2. So make sure to pry those wings apart and take a photo so that we can see the middle of those segments. And then for incidental observations, this is just it's fun. The photo I took there on the left is of Bombus curviellus. That was a fun photo to take for sure. And yeah, you can try it out. Yeah, just to summarize here, we want to capture the bee using insect net, put it into a clean vial, put that vial into the cooler with ice. It'll take 10 to 15 minutes to chill down. They can be there up to 2 hours. Please take those detailed photographs we just talked about and then release the bee within 100 meters of where you captured it. And all you have to do to release them is just sort of, you can put them on an area where you're working and they'll slowly warm up and they'll groom themselves and they'll fly off. It's actually kind of a fun time where you can, watch the bee and get to see it close up and watch some of its unique behaviors you might not otherwise get a chance to see. This is what the data sheet looks like. You can find the data sheet on the website. It's fairly straightforward. There's not a whole lot of detailed information to put on here. The habitat survey that you're doing, all of that information is really what you see here. And it really should be a rapid habitat survey. We're not asking you to spend 4 hours surveying the site. We're just trying to get a snapshot of what that looks like. So site name, grid cell ID if you know it, the date, your latitude and longitude. You can use your phone to find that information, even if you're outside of cell service. If you don't know how to do that, let us know and we can show you how to do that on your phone. How many observers you have, whether it was a point or a roadside survey, the survey start time, end time, size of the, what method you use to capture your bees. Most of you will just be circling all here. And then again, your survey minutes will be 45 minutes for a point survey, 15 minutes for a roadside survey, and then some basic weather information, temperature, cloud cover, wind speed. You can get this information later. You don't necessarily have to get it directly when you're there. If you don't have cell service and you can't use your phone, you don't need to buy a weather device later. You can go home and use a website to check what the nearest weather station to where you were at the time and put that information in there. And then we're going to ask you to survey what you think the habitat type is the dominant habitat type is of where you're doing your survey. It should be one of these 7 options that are displayed here. And then you're going to choose up to 3 surrounding habitats. So if you're in a meadow doing a survey that's surrounded by forest, you might, write woodland forest here as a surrounding habitat type. And then if there's also a farm that's adjacent to where you're surveying or that you can see from your survey area, you might also write agricultural here. You don't have to use 3, but you do have to choose at least 1. This next question, how much of the survey area has flowering resources available? Basically what that question is asking you is, in how much of the survey area could you look for bees on flowers? Was it half of the survey area? Was it 100%? Or was it just really patchy? Was there like one patch of flowers and here and one patch of flowers over there, and you had to walk back and forth between them and you couldn't look for bees in between? That would be like a 10-20 situation, where if there's just kind of flowers, maybe not dense everywhere, but almost everywhere in the plot, you're getting closer to 80 or 90%. It's not a percent cover question. It's more in what percent of the plot could I actively look for bees on flowers? And then just some basic questions about characteristics you saw while you were doing your survey. Did you see bunch grasses, brush piles? And then if you notice or you see evidence of any of these management activities, we just ask that you document them with either yes, no, or suspect. So a really just quick guess. You may not know whether they use insecticides or herbicides, but you might see a bunch of plants that are brown because it looked like somebody sprayed them. So you might put suspect in that case. We're just asking you to do your best with the information that you have. You don't need to spend hours tracking down the land manager and finding this information. And then at the very bottom, you're just going to document your volunteer information, how much time you spent doing your survey, and how many miles that you drove, and you're going to enter this data into the website later. And then on the back, you're gonna enter the plant data. So what we want you to do here is to document the number of flowering plant species that you found in your plot, and then to list the top. Leaf, I feel like this is a picture of the older data sheet and it's changed since then. So I don't want to give people bad information, but I think we're now changed the question so that it is including bumblebees visited, plants visited by flowers, or I'm sorry, it is including flowers visited by bumblebees. And I think we're only getting the top 10 plants. Is that right? I'm sorry. I feel like this is
Yeah,
an
you're
old
right.
slide. Okay.
Yeah, no, you're right. We changed it ever so slightly this year. So you guys can download the current versions from the project resources page on the website.
Okay. All right. So yeah, you're going to just document the plants that you see. Using iNaturalist for this if you're not great at plant ID, it can be really hard, particularly in a place like California that is so diverse with so many different species. iNaturalist or Seek, they have a website. Seek can help you get to at least genus there, or you can post the photo on iNaturalist, and a bunch of botanists will come in and help you identify that to species, and then you can enter the data later. So try to use whatever resources that you have. There's also field guides and apps available to help you identify the plants. But this is really important to help us understand what plants the bumblebees are using and what habitat types that they're using. So please try to do the best you can here. It doesn't mean you need to spend 4 hours, trying to document every species of flower that's out there. We're really just looking for the top 10 most abundant flowers that were out there. and to get them to the lowest taxonomic level that we possibly can. But again, you don't need to spend hours and hours doing this. Do the best you can with the resources you've been given. And then at the bottom is where we're going to enter the bumblebee data. So for each vial that you have, it's going to have a separate row here. You can do your best to identify the species that you had in your vial. We're going to ask you the host plant. You don't have to, though. You could just write bumblebee, and that's fine. You can enter the data that way and let Leaf and I do that work. And then we ask you to enter the host plant. And then here it says photo numbers. And what we want you to do is while you're taking pictures of the bee, we want you to record what the photo numbers are from your camera so that later when you're processing your data at home, you remember which photos go with which bee. So this is an important aspect, and you can write that information here on the data sheet, and then any notes that you might take about that bee. So each vial that you get has a separate line on the data sheet. If you don't happen to find any bees during your survey, that doesn't mean that you just say, oh, I didn't find anything, sorry. We still want and actually need you to submit your data. So you're going to go into Bumblebee Watch and you're going to still submit all of that habitat data that you gathered. And you're going to tell us there's actually a checkbox that you'll see at the bottom of the form that says, I did not see any bees, or this is a null survey. And it's really important that you submit the data and check that box. That null survey information is absolute gold from someone who's trying to assess the extinction risk of animals. If we know that somebody looked for animals and didn't find them in a particular location, that is incredibly important information, and it really helps inform the work that we're doing. So if you do a null survey, please take the time to submit your data. It couldn't be more important. All right, moving on. The next step here is Bumblebee Watch, and you're gonna need an account at Bumblebee Watch to submit your data. So basically what you're going to do is you're just going to take your paper data sheets and you're going to have them on the desk next to you. You're going to go to bumblebeewatch.org and you're going to submit your data. And basically the data forms are set up so that every question on the data sheet has a corresponding form on the website. They're in the same order, and you're just going to follow along and enter the information that you've written on your data sheet into the web form. There's a series of slides here that Leaf has put together, but I'm going to not do what Leaf has asked me to do here. And I'm just going to go to Bumblebee Watch, and I'm going to walk you through the process of submitting a record, just so you can actually see what it looks like. Because I think it's actually it's just more helpful to see it live. And you'll probably see me struggle a little bit as well. And just realize that, like, it's okay. It takes a little while. You may have to figure it out, but it's all going to work out in the end. And it's ultimately not that complicated. So I'm going to start sharing my screen here again. I've navigated over to Bumblebee Watch. And I'm going to see. Okay. Start sharing my screen here. Sorry to buck the trend here, Leaf. I know you're probably like, what is he doing? But I'm just going to do it, and I'm going to go with it. It's going to be great. Okay. Looks to me like you can see Bumblebee Watch here. And so what you're going to do is you're going to go here. If you don't already have an account, you're going to go to the upper right-hand corner, and you're going to click sign in or create an account. And you'll create your account. And once you have your account, you'll get like a profile page where it'll document all of the surveys that you've ever done. You'll get to see all the species that have been verified by you, all the species that you've seen throughout the years. You get to see all the checklists you've ever submitted and all of the bees you've ever submitted on one page. So you can see them and interact with them. You can see them on a map. It's a cool thing to have. It's also, just this past year, we also implemented this new year in review button. So if you click this, you'll get to see summary information. I haven't done anything in 2025 yet, but if I click on 2024, you can see sort of the impact that you had for the program in 2024. So it's a cool new feature that we have on the website. It's fun to look at and see the contributions that you've made over time. When it comes time to submit your data, you're just going to go to add a bumblebee sighting up here in the upper left, and it says select your project. Here is Bumblebee Watch on the left and Bumblebee Atlas here on the right. If you just did an incidental sighting where you took a picture of a bumblebee on a flower, you're just going to select Bumblebee Watch here. But for most of you, hopefully you're going to be doing some survey, and you're going to select Bumblebee Atlas over here. So you're going to select submit sightings here, and it's going to bring up this form. If you're going back to a location where you've already conducted a survey, you can click this button here, choose from your locations, and you can select a location that you've been to before and select that location. So in this case, I'll just pretend that I've done that, and we'll go to, let's go to Ape Canyon. We did it to Ape Canyon. So I select Ape Canyon, and then it automatically will enter the latitude and longitude of that into the database. I don't have to enter any more information. I can just leave it at that. If I didn't go to the same location, but I went to a new location, you can use this part to enter that. You have to give it a site name. And if you don't know the latitude and longitude, but you think you could find it on a map, you can also use the map to figure that out. So you click Show Map. I'm not sure whether it's actually showing this or not. It may not because it's a pop-up, but it'll bring up the map. You can interact with the map and zoom in and click on it. And then once you click on it, I'll go ahead and do it, whether you can see it or not. Once you click on the map, you click Set Location, and it'll automatically enter the latitude and longitude into the database here. And then again, you're just going to follow straight down and answer these questions right off of your data form that you filled out in the form. How accurate is this location? This is telling us how far away could this bumblebee have been from the location that you just entered. Assuming you entered your point sort of in the middle of your hectare plot, we'd like you to select 25 to 50 meters here. That basically would go to all corners of the plot, telling us that that's roughly the error rate of the bumblebees, so that the bumblebees in your survey would be within 50 meters of where you dropped that point. If it's more than that, you can put more. If it's less than that, you can put less. The date of your sighting, again, you can just click the calendar and enter a date. The survey type, point or roadside. We did a point survey. The collection method was I tried to capture all bees. It was 1 hectare because it was a point survey. In this case, it was just me. So it was just one of me. The survey start time, again, you can just enter the time here. So I started at 4:30 in the afternoon, PM, and I went to 5:30 PM. You'll notice that this is an hour here, right? This is an hour, but my survey time here, I'm going to put 45 minutes. The reason that this is an hour and this is 45 minutes is that the hour was the whole time it took me to do my survey. So I spent 15 minutes then transferring my bees and putting them in the cooler and taking notes and doing all that other stuff. So that's why the survey start and end time don't necessarily match up with the survey length, which should always be 45 minutes if you're doing a point survey and 15 minutes if you're doing a roadside survey. You're going to enter the temperature. It was a lovely 68 degrees. There were no clouds that day. No, I don't need to enter any weather notes. I could if I wanted to. It rained for 10 minutes in the middle of my survey, so I paused it. That'd be a fine thing to do. The habitat type of my survey was I was in shrub scrub, and I was surrounded by grassland meadow beyond that. And then there was also some forest nearby. So I'm going to enter those two. How much of the survey plot could I actively search for bumblebees on flowers? It was about 50%. Brush piles, there were some brush piles and some mulch and some rodent holes, but I didn't see those other features. I didn't see any evidence of mowing, no evidence of grazing, but I did see some evidence of native grazing, so I'm going to suspect that. No agriculture, no insecticides. I did see some herbicides, or what I thought, so I'm going to say yes. I'm going to suspect there was a fire nearby because there was some scarred plants. And then I did see some honeybees, so I'm going to suspect that. If there were any management notes, like how many honeybee hives you saw, or you know that somebody sprayed pesticides or whatever, you could enter those in here, but you don't need to. And then it's time to enter your flower species. So let's say I saw one flower species just for ease of entering data and for time. We have a new feature this year on the website where we're actually integrating with an external database to try to standardize our plant names. So if you, let's say, caught this plant on a lupin and you just start typing, lupin in here, you can see that it'll quickly filter it down and give you a whole host of options to choose from. This is the plant that I caught this on is Lupinus lepidus. And you can see if I just start sort of typing in here, that may not come up, right? But if I keep typing and I'm persistent and I type lupinus, eventually you'll see that the species that I want pops in. If you wanted to learn more about it, you could click on the info here and it would actually open up a profile of that page of this flower and tell you more about it. But in this case, I know it's this. I'm going to select it. It's going to enter that, and now it's entered. In this case, I get paid to work for Xerces, so it's not a volunteer effort. But if it was, I'd click this and it would open up some questions that you would have answered at the bottom of your sheet that you're entered. In this case, you're using an online resource, so you're going to say yes here, and you're going to then say that this was on April 9th, 2025. In this case, I'm just going to check no here. And then in this case, I'm just going to submit this well, no, I won't. I'll go to the next page. I'm going to say that, yes, I did submit bees and detect bees in my survey, but if I didn't, this is where you would tell us that this was a null survey, and then you'd be done. But in this case, I'll say yes, and then I'm just going to type my name here saying that I entered this correctly. And then I'm going to proceed. Oh, and it warns me, you didn't name your site, Rich, so you have to enter a new site. So I'm going to say just test California. And then, so it warned me that I couldn't go on unless I named it, which is helpful. Oh, there's something else that I didn't enter. I didn't enter the wind speed. So I needed to enter that the wind was 6 to 10 miles per hour. And now I'm going to go and I'm going to proceed. And now it's going to let me go to the next site. Now, for each of those lines, now it's time to enter your bumblebee data. And so what you're going to do for each bumblebee, you're going to click Add Sighting. And then here you're going to be able to either drop the photos or select the photos. You can choose the species that you think you found here. If you want, you can use our identification tool, which is a pop-up where you can say this bee had a yellow face and it had yellow at the front and it had a yellow stripe on its abdomen. And then it's going to narrow down like which of the 4 species in California that it could have been. And then you could select one of those and click Vosnesenskii and select species, and it will enter the species information in there. You may not see this count field. This is something that I have because I'm special. You probably won't see this in your data form. But then again, if you want to, guess at the sex of the bee, if you think it was a queen or whatever, you can enter that here, or you can just leave it as not determined. And here, once again, you're going to enter the floral host. So, same thing, Lupinus lepidus. Just keep typing, it'll pop up, you select it, and then if you had any other notes, you could add them here. And then you click Add Sighting, and it'll add this sighting to the database. One note, particularly for Franklin's bumblebee, for those of you surveying in Northern California, Franklin's bumblebee is an endangered species. And any record that you think might be Franklin's bumblebee needs to be marked as private. So please use this private to keep it obscured from the general public if you need to. And then you'll click Add Sighting. It's not going to let me do that because I didn't upload any photos, but then you'd go back and you'd click Add Another Sighting and keep adding sightings to your checklist until you're done entering all the bumblebees that you detected. So that's how we entered data in Bumblebee Watch. And I think more or less the end of the information that Leaf had wanted me to share with you. So I'm going to say there's a few more summary information. There's a couple of resource slides here. I guess the one thing that I will say, I guess I'll put up these I will put up the environmental compliance slides for you to see because it's important that we share this information with you. But it is really important that in order to do any of these surveys, you need to be on our scientific collecting permit and have a copy of our MOU, our Memorandum of Understanding. In order to get on those, you need to take a quiz. And the quiz, we can share the link to the quiz, but the quiz basically shows that you know how to safely handle bumblebees. When we send you those scientific collecting permits, they have conditions in them. And please read those conditions and please follow those conditions. They are going to allow us to keep doing the work that we're doing. If we have volunteers that don't follow the conditions, that don't see some of the things on this slide here and follow through with them about bald and golden eagle protection, about rabbit hemorrhage disease, we're going to lose our scientific collecting permit, which would restrict the ability for us to gather this information. So please take this seriously. Follow the regulations that CDFW has set aside for us. They've been very generous in setting up this project for volunteers. They did this for us, allowing us to add named individuals that have passed this quiz. So please take it seriously. And also recognize that once you have a scientific collecting permit, that is only valid for participation in the California Bumble Bee Atlas. It does not give you permission to do other survey work, either for fun or for work. It only gives you permission to participate in the California Bumble Bee Atlas. So please keep that in mind. If you need a scientific collecting permit for your own work, for your own consulting work, or for some other survey that you're doing, you need to find your own scientific collecting permit and talk to CDFW about the process for making that happen. There's a video you'll have to watch in order to take the quiz, so please watch that video. It'll talk about the bald and golden eagle act, and as well as rabbit hemorrhage disease and some of the sensitive habitats to avoid. I talked some about Franklin's bumblebee. This is the general survey area on the right here, Northern California, Southern Oregon, where you may detect that species. Please take this very seriously. When you upload it to Bumblebee Watch, please mark it as private. And then we've also been asked by the U.S. Fish and Wildlife Service to not share this finding on social media, to not publicize it at all. This species has not been seen since 2006, and we don't want to love it to death. So please, please keep that in mind. Okay, I'm going to stop. Leaf, I'm sorry I took too long, but that's what you get when you invite me to help you, I guess. I'm sorry I went over. But yeah, that's a lot of material to get through. So thanks a lot.
Yeah. We are running a little bit late, so I'm sorry for this, but I'm gonna cancel our 3-minute break and just move right into the final slide deck, which will take about 20 to 25 minutes. So I'm sorry we'll be over just a little bit. If you have to leave, we understand, and I would advise you to watch the last 10 minutes or whatever it is on the pre-recorded version that we will have, that we do have linked on the website under the Project Videos tab. So let's get into identification. And as you'll see, we were already not going to teach you to identify all 25 species because we simply don't have time to do that in this format. We will be likely offering some ID webinars later in summer, likely, not sure yet, but those are excellent places to go deeper on this stuff. So we're going to start by talking about, I'm going to just skip this slide. We can talk a little bit about morphology and how to distinguish, to decide if it's even a bee, and then is it a bumblebee as opposed to some other kind of bee, and then is it male or female, and so on. And then we will look at the species of greatest conservation need species and their lookalikes, and that's the extent of what we can do tonight with ID. So here is that diagram of the various morphological and hair characteristics. You heard Rich talk about T's or tergal segments, and we'll be talking a little bit more about that, T1 through T6 or 7. There's a pollen basket on the hind leg. We've talked a bit about the various parts of the body, but the head includes what we call the face, which is the vertical area between the compound eyes where the antenna emerge. And the vertex, which is the top of the head near the simple eyes, that's another place we find hair color that's helpful in identifying the bees. There's some other stuff in the text here. The underside of the abdomen can be important. The cheek that Rich described is really important to ID. Here is a better look at the cheek. This is the rusty patch bumblebee, not one that we have in California. And the two arrows are showing the two dimensions of the cheek, so length and width. And in this case, the width is longer, slightly longer than the length. And that is an important distinguisher for the bumblebees in this subgenus, in this category. So we really need a nice photo that shows that cheek. Okay, so we're going to use this flow chart, and I'll go through this fairly fast in the interest of time. But let's imagine that we have a mystery insect, and we want to know, is it even a bee to start with? And I've suggested a couple of resources on the side for at various stages. So we'll figure out, is it a bee? Is it a bumblebee? Is it male or female? And finally, is it a cuckoo, or is it one of the other types? So first of all, in terms of insects in general, these photos probably all look a bit like bumblebees, or sorry, like bees, um, or maybe some of them don't. But people are often confused by what a bee is. And so if you are taking a guess in your head right now, here are the answers. Only one of these is actually a bee. None of them are bumblebees. All of them are flies, except for the one in the upper left, which is a solitary bee, a parasitic bee called nomada. And so many flies closely mimic bumblebees. They're large and robust and hairy. So much so that this book called Bees of the World, which sounds pretty authoritative, in fact it is, got a cover photo of a fly rather than a bee. Thankfully, in the second edition, they corrected that with a nice bumblebee photo. So if you find this stuff confusing, just realize that Christopher O'Toole and Anthony Raw also have a hard time with it, and they are experts. So here are some characteristics of what a bee is. Bees evolved from wasp-like ancestors, and the ancestor of both bees and wasps was a meat eater. It fed animal protein to its larvae as the protein source. But bees do it differently. They are vegetarians who feed on flowers, and so that's the key distinction about bees, and they have a number of adaptations associated with pollen harvest and also nectar harvest. So long tongues for the nectar, structures for carrying pollen like you see on the underside of the bee on the left or on the hind leg of the bumblebee on the right. They have branched hairs, as you can see in that image. They're usually not visible to the naked eye, but if you use a hand lens, you can indeed see them. So we're going to assume that you can figure out that it's a bee based on the fact that it's harvesting pollen and carrying pollen and some of these other characteristics. Now, let's try to figure out, is it a bumblebee? And so here are some species that look like bumblebees, but are actually not bumblebees that you can see in California. The oil bee on the left, carpenter bees, which I think everybody's going to have an idea of, a conception of already. Honeybees, of course, and digger bees, this genus Anthophora. There are other large, robust, hairy bees in California that look like bumblebees, but these are some of the more common ones. So honeybees are honeybees. We've talked a little bit about them, and they look distinctly different from bumblebees in terms of being kind of honey-colored, not a lot of hair on the body, and being sort of torpedo-shaped. That's often how they're described. The other bees in this image, they are carrying pollen on the hind leg, but you'll notice there is no barren, bare, shiny area where they're placing the pollen. Instead, it is a thicket of hairs. It's a brush, and they're packing dry pollen into that brush. And this is a key distinction between female bees that are that look a lot like bumblebees and bumblebees themselves. And bumblebees are going to carry the pollen more like that honeybee right there. So here are some photos of bumblebees doing their thing. So they're large and very hairy in almost all cases. In California, they will almost always be dominated by black and yellow coloration, but they may also have various other colors on the body. And they have that characteristic pollen basket if they are female and not a cuckoo bumblebee. So we'll make the assumption that we know it's a bumblebee, and now we want to know, is it male or female? This is actually, I think this is probably the easiest of the several different levels that I'm talking about here. So bumblebees, males and females differ in some basic ways. Males have 7 abdominal segments as opposed to 6. Um, the males, obviously, they lack the pollen basket or corbicula, which is circled in red there. So instead, they just have a leg segment. It can be a little bit dilated or widened, but it's not going to be concave. It's not going to be shiny and hairless. It may have a little bit of shine, but it's distinctly different from what the female has. Um, Males have one extra antennal segment, so 13 instead of 12. Oops. Um, Males cannot sting. They do not have a stinger. The stinger is actually an ovipositor, and so obviously only the females lay eggs, and males are incapable of stinging you. Males are often shaggier. They can be a little smaller, or a little bit larger than the workers. It depends. Some of them have markedly larger eyes than the females, and the color patterns can be just identical, or they can be a little bit different, but typically they're going to be somewhat similar, as in this species here, Grisiocaulis, which occurs in Northern California, only in California. Occurs broadly across the continent otherwise, but the color patterns are largely concordant there. All right, the last one, which is actually a little bit tricky and takes a little experience, we are going to use the hind leg tibia to decide if it is a cuckoo or Cytherus, or it's one of the non-cuckoo or non-Cytherus. And we'll divide that by males and females here. So here are the 4 looks, and those are just thumbnails. We'll look at them in like this, zoomed. So Cytherus males, the hind leg tibia, that place where the pollen is carried by the females of non-cuckoo species, it is convex or somewhat flattened, is typically not very shiny, and it's typically covered in rugose like bumps. You can kind of see that in this image, these bumps everywhere. The hairs around the edge of the segment are not very robust. Those yellowy hairs, they're not big enough to arc over the top and hold pollen down. He's male and he's also Cytherus. So this one looks the least like a pollen basket. Here is what males that are of normal non-cuckoo bumblebee species look like. You can see that little shiny elliptical area on the segment in the middle, but you can see it's also surrounded by a bunch of rugosity or bumpiness, and the hairs on the side of the segment are also not very robust. We will compare those hairs to the final one, the 4th one here. So here's what a Cytherus or cuckoo bumblebee female looks like. It is dilated, right? It's a wider body segment, and you might initially think that that was a regular non-cuckoo bumblebee female, but in fact, if you look at that thing, it is bumpy all over. There's some shine, but it is not flat and shiny. It is, in fact, not concave. It's got a little convexity to it, and again, the bristles on the side of the segment of the leg are not particularly robust. Here's the one with the robust segment, the robust hairs. So look at how strong, how thick and long those hairs are, and they arc right over the pollen mass. Also, you can see, you don't see that rugosity or bumpiness. This is a shiny, hairless and slightly concave area on the non-cuckoo bumblebee females. So we have this ID guide. This is a good place to begin if you're a beginner at bumblebee identification. It's downloadable on the website, and it's also in the project manual. This is 1 or 2 images of every female bumblebee species in the state. And I've categorized them by gross color pattern for the most part. So you see on the left, number 1, those are bees that have orange or red hair somewhere on the body, anywhere. Next, it's yellow and black bees with a yellow stripe on the first abdominal segment. Next, yellow and black bees with a black T1 or first abdominal. And then the next ones, those 4 species have white hair somewhere on the body. And finally, the 3 cuckoo bumblebees. This isn't easy at first, but this is where you start to identify bumblebees. There are lots of other identification resources out there, but we'll start with this little thing. And I'm now showing you the species that I'm going to show you in detail. These are just our species of greatest conservation need in California, and we won't have, as I said, won't have time to go through all of the other ones. So starting with Bombus franklini, or Franklin's bumblebee, this is our rarest species. It occurs only where those orange dots are on the California map, and in adjacent Oregon. And the entire global range is indicated in the little inset map, just the black dots in Northern California and Southern Oregon. So tiny little range. This species is listed as endangered by Fish and Wildlife and is a candidate for listing in California under CISA. We have not seen this bee since 2006. It could be extinct. We are trying to find it, and we so far have not been able to. The purple arrows are showing you where the field marks are on this bee. The one is on the face. It is a short cheek, and you obviously can't see that in this photo, but we don't actually have a lot of photos of this species. So a short cheek, shorter than wide, wider than long. On the thorax, you're getting yellow hair, and it forms this characteristic little horseshoe pattern right in the center of the thorax between the wing bases. It'll take a little practice, but it is a horseshoe shape, and that is the only species in California that has something like that. It then has some yellow hair sparingly on the distal abdominal segments, T4, 5, and or 6, but just a tiny bit of pale hair, not very much at all, as compared to Western bumblebee and some others that we'll take a look at in just a second. Okay, here's Western bumblebee. The range map shows, now it's showing you the orange dots are older records and the blue are more recent ones. I need to update these slides, obviously, because we have more data now. This bee looks a lot like Franklin's, and it's also one of our species of greatest conservation need. They're close relatives, which explains the similarity. This species has a short cheek also, and you can partially see it in this image. It's generally going to have a black patch of hair across the middle of the thorax, as indicated there, and then varying amounts of yellow hair at the front of the thorax and varying amounts of yellow hair at the back of the thorax, often without. And then as you can see in the little graphical images, the thorax varies quite a bit. Sorry, the abdomen varies quite a bit in its hair color, but there is always pale hair at the tip of the abdomen. It's salmon colored in this image, in this photo, but it's often white. This species has disappeared from the California coast with almost entirely, and is now found in California. Now we're only finding it in the northern Sierras. And we hope you will help us find it in the Bay Area or elsewhere in its former range. So there are three species of what we call yellow-faced bumblebees that look similar to those two, or grossly similar. One of them is of conservation concern, a species of greatest conservation need. That's the first one in this picture in purple, Bombus calligenosus. It is also known as fogbelt bumblebee. It is a black body with yellow facial hair, yellow shoulder hair, if you will, on the front of the thorax, and then a single yellow stripe on the fourth abdominal segment. Vosnesensky at the bottom of the set of images, identical to what I just described, and Van Dykii in the middle, identical, except the yellow stripe is on the third rather than the fourth abdominal segment. And the range map is showing points for all three species. Only the first one, calligenosus, is rare or uncommon in California and is really restricted to coastal areas. There are a few mistake observations on that map, unfortunately. But these guys can be mistaken for Franklin's or for occidentalis or Western, just by gross color pattern similarity. This is Bombus furvidus, also can be mistaken for one of those species in California, in that it is largely black with yellow in most of the same places. And sometimes that yellow on the abdomen will look like the pale hair at the tip of the tail, the white hair I was just talking about for Western bumblebee. This guy is very distinctive once you get to know it. It's also known as California bumblebee or Bombus californicus. It has black facial hair. The others that we just looked at had yellow facial hair. So look at that head. It is all black. There can be a minority of yellow hairs mixed in there, but usually it is predominantly black or entirely black. Same general color pattern with the yellow stripe on T4 and the yellow shoulders. But the other thing about this one is it has a very long face, much longer than those other species that we've looked at so far. And so you need to get a look at the cheek for this one, and then you will immediately see it is just not in the same category as the others that we've looked at in the slides here. Okay, one more that can be mistaken for these species we've looked at is Bombus sitkensis. It's quite distinctively different, but the thing in common is the pale hair at the tip of the tail. It's a kind of a salmon color, yellowy, dingy, off-white. It can sometimes be kind of orange. The bee has a lot of black hair on the thorax and on the face. It has a very dusky, dark thorax look to it, and it's quite hairy, long hair. It's not all that similar to Western bumblebee, for example, but for beginners, this one sometimes trips them up. It has a cheek that is just longer than wide, so as opposed to shorter than wide, as we've discussed with Western and Franklin. And I'm showing all that mix of black hair into the yellow hair on the thorax. Um, there is yellow on the first abdominal segment. Okay, let's talk about Crotch's bumblebee. And this one is the one that everybody's interested in because it's a species of conservation concern. It's provisionally protected as a candidate for listing under CISA. It is endemic to California. We have a single Oregon record, a single Nevada record, and about 50 Baja California records, and all the rest of them come from California. It should be our state insect. If I were king, it would be. It's a special bee. It's large, robust, just beautiful, short, even hair. It's sort of velvety in some light. This is a species with a short cheek, distinctively short. The vertex is going to have a mix. It's going to have yellow hair, or sometimes a mix of yellow and brownish black hair, as you can see on this bee. Um, the tip of the tail, and about 3/4 of them is going to be red, and the other quarter it's black. This bee is distinct, and just once you get to know it, this is an easy one to learn for beginners. Here are a few that look similar to it. So this is red-belted bumblebee. It's the most variable in its coat color of any of our bumblebees. And you can see from the little thumbnail sketches that it can look grossly similar. It can have a red tail. It has a short cheek also. This is a species, however, that mostly does not co-occur with crotches, except in the Bay Area. And so when you find this guy in the Eastern Sierras, you're probably not going to have to consider crotchii in most cases. So that one can be ruled out in most places. This one is also mainly in the Eastern Sierras and Northeastern California, but it can be found broadly in other spots. It's a large, robust bee with short hair. Generally, most populations do not look like Crotch's bumblebee. They're largely yellow with a strong component of black towards the tip of the tail with black facial hair. However, in California, in two places, that would be Humboldt County and San Miguel Island, there are bees of this species that have that red tail and that pattern in the little graphical images that is just identical to Crotch's bumblebee. The only distinction that you will notice is this one has a long cheek and Crotch's has a short cheek. So if you're in Humboldt County, keep an eye out for this one. Otherwise, you probably don't have to worry about it being a lookalike for Crotch's. Finally, Suckly's bumblebee. This is our other species of greatest conservation need, and it's one that we're searching hard for in Northern California. We've only seen it 4 or 5 times ever in the state, and it's mostly a bee found in more northerly places, and it's currently common in Canada, but not anywhere, particularly in the Rockies in the US. This is a cuckoo bumblebee. It can be distinguished from other cuckoos in that it has a lot of yellow hair on the side of the thorax, which that arrow is pointing to. There are some distinctions about the underside of its abdomen, at the end of the abdomen, the shape of the abdominal segments. I'm going to not get into the detail about that right now. This is a very rare bee that you're only going to see in Northern California if you find it. And if you think you might have found it, we definitely want to hear about it. Make sure that you submit that data. So that's all I can say about that one for now. This is a common cuckoo bumblebee species that is often mistaken for the one we just looked at. It typically, the one way that's easy to distinguish it, it has a patch of yellow hair on the face. And you can see that in this image. Some of them is a lot less obvious than this bee, but they will always have at least a few yellow hairs right between the eyes in a patch. And Suckly does not have that. It has black facial hair. And with that, I'm going to say thank you for your patience with us and sticking around until now. And we're just very appreciative that you are choosing to join the project. We think this is really important work that needs to be done. And as we've both told you separately, you are the best people to do it along with us. We can't do this alone, and we greatly value the contributions that you're going to make to the project. So I don't think we have time to answer questions. I will follow up with any that need answering by direct email to all of you, or maybe just to the individual person if it seems like it's of interest only to them. So please get in touch if you have further questions, and please remember that there is a quiz for you to take It's linked on the website if you need to find the link. After that, you'll be permitted and you'll be ready to start collecting data with us. So thank you very much.
I think I got to most of the questions, Leaf. I will drop
Great.
a link to the quiz in the chat here in case folks
Wonderful.
need to find it. This tiny URL that you made works.
Yeah, it does.
Okay. Yeah, I see a question there about when you need to take the quiz. The answer to that is you need to take the quiz before you do surveys. So if you pass the quiz, you should automatically get an email with us with attachments to it, that'll include the scientific collecting permit, the MOU, and the associated conditions. And then you'll be free to go do surveys, and that should be an immediate process. If it's relatively immediate, I don't know if it runs like immediately or hourly or something like that, but you should get a response within a day. Let's just give us that much freedom, I guess. If you don't, feel free to reach out and say, hey, I didn't get mine. It could be that you typed your email wrong, or that our scenario is broken for one reason or another. So it helps us if you didn't get it. You should, but that'll trigger us to go look and figure out what's going on.
Right. Okay, well, thanks everybody. Have a good evening. And again, get in touch with questions and we will see you hopefully at a field event.